Introduction. Welcome, students, to today's lecture on purine and pyrimidine metabolism. In this comprehensive script, we will delve into the intricate world of nucleotide biosynthesis, catabolism, and their various implications in health and disease. Before we embark on this journey, let's briefly touch upon the fundamental concepts of purine and pyrimidine metabolism. Purine and pyrimidine nucleotides serve as the building blocks of DNA, RNA, and essential coenzymes involved in various cellular processes. As medical professionals, it is crucial for us to understand the biosynthetic pathways, regulation, and dysfunction of nucleotide metabolism, as it relates to several genetic disorders and potential therapeutic interventions. Now, let us proceed to the first topic of our discussion. In this section, we will explore the fundamentals of purine and pyrimidine metabolism. Purines, such as adenine and guanine, consist of a bicyclic structure fused to a 5-membered imidazole ring, while pyrimidines, like cytosine, thymine, and uracil, possess a single 6-membered ring structure. Within the human body, purine nucleotides are synthesized via Denovo pathways, whereas pyrimidines can be obtained through both Denovo Synthesis and Salvage Pathways. We will discuss these pathways in greater detail in subsequent topics. Understanding the intricacies of nucleotide structure, function, and metabolism is vital for comprehending genetic diseases associated with impaired purine and pyrimidine metabolism. Therefore, let us progress to our next topic for a closer look at enzyme kinetics involved in these pathways. Enzyme kinetics is a branch of biochemistry that examines the rate at which enzymatic reactions occur. In purine and pyrimidine metabolism, several enzymes play crucial roles in catalyzing different steps within the biosynthetic and catabolic pathways. By studying enzyme kinetics, we can better grasp the regulatory mechanisms governing nucleotide synthesis and degradation. Furthermore, understanding the properties and kinetics of these enzymes aids in the development of pharmacological interventions to target specific metabolic pathways. Now that we have established the significance of enzyme kinetics in purine and pyrimidine metabolism, let us delve deeper into the structure and function of nucleotides themselves. Nucleotides are composed of 3 main components: a nitrogenous base, a 5-carbon sugar, ribose or deoxyribose, and 1 or more phosphate groups. The variations in these components give rise to the diverse functions of nucleotides in various biological processes. In this section, we will explore the structural differences between purine and pyrimidine nucleotides and discuss their respective roles in DNA and RNA synthesis. Additionally, we will delve into the functions of nucleotide derivatives, including energy currency molecules, for example, ATP, GTP, coenzymes, for example, NAD, FAD, and second messengers, for example, CAMP. With a solid understanding of nucleotide structure and function, we can now proceed to uncover the intricacies of Denovo synthesis of purine and pyrimidine nucleotides. Denovo synthesis is the primary pathway responsible for the de novo biosynthesis of nucleotides. This complex process involves multiple enzymatic reactions, which take place in different cellular compartments. In this segment, we will meticulously dissect the individual steps involved in the de novo synthesis of purine and pyrimidine nucleotides. We will delve into the regulation and key enzymatic reactions governing the production of inosine monophosphate, IMP, and orotidine monophosphate, OMP, the precursor molecules for purine and pyrimidine biosynthesis, respectively. Understanding the de novo synthesis pathways is essential, but so is being cognizant of alternative routes available to the cell for nucleotide synthesis. In our next topic, we will explore the salvage pathways in purine and pyrimidine metabolism. In addition to de novo synthesis, cells possess salvage pathways that allow for the recycling of nucleotides from degradation products or from the surrounding environment. These pathways enable efficient utilization of nucleotides and help maintain the delicate balance required for normal cellular function. In this section, we will investigate the enzymes and reactions involved in the salvage pathways for both purines and pyrimidines. We will explore how salvage pathways act as a crucial mechanism for nucleotide synthesis, particularly in situations where de novo synthesis may be compromised. As we continue our exploration of nucleotide metabolism, it is integral to comprehend the catabolic processes that balance synthesis and degradation. Let's move on to our next topic, purine and pyrimidine catabolism. Purine and pyrimidine catabolism refers to the breakdown of nucleotides into simpler molecules. The end products of catabolism are excreted from the body or recycled into de novo synthesis pathways. This section will focus on the enzymes responsible for breaking down purine and pyrimidine nucleotides, resulting in the generation of uric acid and other byproducts. We will also discuss the significance of purine and pyrimidine degradation in maintaining cellular homeostasis and the clinical implications when these pathways are disrupted. Speaking of disruptions, deficiencies in purine salvage enzymes can lead to severe genetic diseases. Let us now explore the disorders associated with purine salvage enzyme deficiencies in our next topic. Purine salvage enzymes are vital for the recycling of nucleotides, ensuring the availability of purine building blocks for DNA and RNA synthesis. Deficiencies in these enzymes can give rise to a spectrum of genetic diseases, such as Lesch-Nyhan syndrome and adenosine deaminase deficiency. In this segment, we will investigate the underlying mechanisms and clinical manifestations of purine salvage enzyme deficiencies. Additionally, we will explore potential therapeutic strategies aimed at ameliorating symptoms and improving the quality of life for affected individuals. Now that we have extensively covered genetic disorders related to purine and pyrimidine metabolism, let us shift our focus towards the pharmacological interventions targeting these metabolic pathways. Pharmacological interventions play a crucial role in managing disorders arising from imbalances in purine and pyrimidine metabolism. It is essential for us, as medical professionals, to be aware of the pharmacological agents used to modulate these pathways and their potential therapeutic benefits. In this section, we will explore specific drugs targeting enzymes involved in nucleotide biosynthesis and degradation. We will discuss their mechanisms of action, clinical applications, and potential side effects, providing you with a comprehensive understanding of drug interventions within purine and pyrimidine metabolism. As we progress through our lecture, let us now direct our attention towards the intricate regulation of nucleotide synthesis in specific organs and cell types. The regulation of nucleotide synthesis is tightly controlled, ensuring that the production of nucleotides meets the specific demands of different organs and cell types. This differential regulation allows cells to adapt to varying physiological and pathological conditions. In this section, we will explore the tissue-specific regulation of nucleotide synthesis pathways. We will discuss how these regulatory mechanisms are influenced by metabolic needs, hormone signaling, and genetic factors. Moving forward, it is crucial to expand our horizons and explore the impact of purine and pyrimidine metabolism in microbial pathogens. Purine and pyrimidine metabolism is not limited to human cells alone but also impacts microbial pathogens. Understanding the metabolic pathways utilized by pathogens aids in the identification of potential therapeutic targets for combating infectious diseases. In this section, we will examine the similarities and differences in nucleotide metabolism between human cells and microbial pathogens. We will discuss how targeting specific enzymes and pathways can lead to the development of antimicrobial agents. Now, let us shift our focus to the role of nucleotide-based signaling molecules in inflammation and immune responses. Beyond their role as building blocks, nucleotides also serve as potent signaling molecules, modulating various physiological processes, including inflammation and immune responses. In this segment, we will explore the function of nucleotide-based signaling molecules, such as adenosine triphosphate, ATP, and cyclic adenosine monophosphate, CAMP, in the regulation of immune cell functions, inflammation, and host pathogen interactions. Lastly, let us wrap up our comprehensive discussion by exploring the genomic consequences of defects in purine and pyrimidine metabolism. Deficiencies or dysregulation in purine and pyrimidine metabolism can have profound effects on genomic stability and function. This topic examines the genomic consequences of defects in nucleotide metabolism, including mutations, DNA damage, and imbalances in nucleotide pools. We will discuss the implications of these genomic alterations for cellular processes, such as DNA replication, repair, and gene expression, as well as their potential role in the pathogenesis of various diseases. In conclusion, understanding the intricate world of purine and pyrimidine metabolism is crucial for medical professionals. The biosynthesis, catabolism, and regulation of nucleotides play pivotal roles in maintaining cellular homeostasis. Moreover, dysregulation of these pathways can contribute to the development of genetic diseases and may offer potential targets for therapeutic interventions. Throughout this lecture, we have explored the fundamentals of purine and pyrimidine metabolism, including enzyme kinetics, nucleotide structure and function, Denovo synthesis, salvage pathways, catabolism, genetic diseases, pharmacological interventions, regulation, microbial metabolism, nucleotide-based signaling, and genomic consequences. I hope that this comprehensive script has provided you with a solid foundation in purine and pyrimidine metabolism. Remember, the knowledge gained from this lecture will be of great value in your future medical careers. Thank you for your attention, and I look forward to our future discussions on complex topics in biochemistry.